in this talk, I'm going to show you an approach based on deep learning to detect and segment 3D images. I will focus on two typical projects. First, we will study images acquired with a very sophisticated microscope in cryo-electron tomography. I guess that you are not so familiar with this technology, which is can produce very spectacular results, but also represent serious challenges in image processing. And then we will show you that this methodology can be adapted to detect very rare events, exocytosis events observed in a turf microscopy. So for people who didn't attend the lecture on last Friday, I summarized the convolutional neural network on 2 slides. And the idea of a convolutional neural networks introduced now 10 years ago or 15 years ago can be decomposed into 2 parts. First, you have the feature extraction. In this stage, the idea is to apply very basic convolution and polling operations to generate a low-dimensional representation of your input image. And this vector, the feature vector, is generally used to feed a neural network for image segmentation, as you can see on this slide. Right. The most popular architecture is a UNet architecture, which apply different subsampling and upsampling operations plus additional connections. And for the user needs just to submit a training data set composed of input image and target images. And the target images are segmented images in general. So you have binary mask to train the model. Right. So you need a lot of data to train the model parameters. The model parameters are the neural network parameters plus the weights of different filters that I just mentioned before. Okay. And so since the introduction of AlexNet, there was very successful algorithm for image segmentation and image classification for 2D image analysis. For instance, you have faster RCNN can detect and label objects. And the most popular method that you know probably is cell pose. I guess you played with these tools, especially cell pose and maybe SAM during the different workshops during this event. So don't forget that cell pose, which is was trained from very large data sets and in general based on UNet architecture as well. And now we will see in the future if SAM microsum can do a better job, but this SAM is based on the new technology based on transformers, which is the new generations of artificial intelligence methodology. Okay, so these methodologies were very successful to segment 2D images, but there are not so many tools for 3D image analysis. So now I'm going to focus on another technology, which is not fluorescence microscopy, but cryo-electron tomography, which is not a cheap system, but the system has the advantage that you can observe all macromolecules in intact cells in situ. You mean that you have the ability to detect maybe 100 macromolecules, maybe more, in the cell, provided you can detect these macromolecules with very small objects in general. The pixel size in that case is 3 angstrom in general. So this is a very high separation technique, 0.3 nanometers in general, and you get 3D volume from tilt series here. So from the different tilt series, you apply a reconstruction algorithm to generate 3D volume composed of several hundreds of voxels. Right. The problem is that it's not so easy to detect macromolecules in such images. For instance, let's just consider the larger macromolecule that you can detect by your eyes is a ribosome. A ribosome are generally located in the cell. They are close to the membrane, or they are free in the cytosol. And the first problem that we have to solve is to detect these ribosomes in 3D volumes, and you see it's not so easy to detect such complex data. But this macromolecule is quite large compared to other macromolecules. So to give you an idea of the problem, I'll show you here a few sub-3D tomograms. And the quiz is to detect who is who in this, and these are 16 sub-tomograms. So where are the background proteasome, the ribosome bound to the membrane, and the ribosome? I guess no one here is an expert to detect who is who in this subtomogram. So you need experts to label such subtomograms, right? And the answer is the following one. For maybe some of you saw my talk a few months ago, but even if you know that solution is not easy to do, how can you detect such macromolecules in so complex data? The data are corrupted by a missing wedge, noise, and so on. That's why it's very hard to detect. And as in fluorescent microscopy, you have to reduce the dose of electrons to preserve the specimen as well. That's why you have very noisy image to analyze in that case. So you need experts. And in general, the number of experts is not so high in the literature. So it's not cheap, right? But I guess you don't need to be an expert to detect cars in a picture. I guess your schedule can do that very easily. So you can collect many annotations from these pictures, and then you can train more easily system to detect cars in multimedia videos in general. So this is a challenge, right? So that's why we have a collaboration with Max Planck Institute, who they supply annotations collected from maybe 6 years of it's a campaign of 6 years annotation, right? Actually, the PhD students labeled and annotate different macromolecules and collected data sets, which become in the future a training data set for us. And they told us, can we design a 3D neural networks able to perform as well as the human expert, to save time, of course, and to be more robust if possible. So in that case, we bought a few GPU cards for 3D channel learning, and so the idea is the following one. We have only 3D coordinates of macromolecules, so you have no segmentation. You have just the 3D position of macromolecules with an ID of each macromolecule. And so because neural networks are based on image segmentation, we have to convert the design notation into segmentation map. And at the end, from the segmentation map produced by the neural network, we have to convert this segmentation map into 3D coordinates. Right. Just an information, in general, the template matching is used to do this job. It takes maybe several hours to process a 3D tomogram. And each template matching is dedicated to one macromolecule, so you have to run the template matching several times. So it took a few hours, few days to process a tomogram. See, in that case here, our motivation was to detect several macromolecules in one run. Okay. First, when you try to apply the technique generally developed for 2D images, you have a few problems. First, you have a problem of memory, right? It's not possible to feed a cellular tomogram to the network, and so we have to select a few 3D patches for training. Second, we have a problem of imbalance. Actually, only 0.5% of voxels are labeled as macromolecules. So, in general, the system needs to tell you that there is nothing in the 3D tomogram. And so we designed a patch sampling procedure that contained only one macromolecule pixel for training. Okay. And so we use this 3D patches for training. But I said that the expert only provide 3D coordinates, X, Y, Z. And the idea is to convert these 3D coordinates into segmentation map. It's not an easy task. And to do that, actually, we apply the usual technology used in cryo-electron tomography. From 3D subtomograms, we compute a subtomogram averaging, which consists to align and compute an average of all these macromolecules. And then at the end, you have an average of a 3D average of the macromolecules. And we threshold this image to get a mask. And this mask was repositioned at the 3D coordinates given by the expert. So we have now target images for training. So we supply this image and the target images for training. Right? Given this, so it takes a few days to train the model, but at the end we have it takes a long time for training. But at the inference stage, it's much faster than previous methodology. So during the inference stage, you provide you supply the 3D tomogram. You apply you collect the output of the neural network, and at the end you convert these segmentation maps into 3D coordinates by applying a clustering algorithm. It could be a K-means algorithm or mean shift algorithm in that case. Right, so it worked. It took maybe 5 years to develop these technologies, but it was quite successful in different challenges. In the community in cryo-electron tomography, laboratories designed several shred challenges for 3 years. And you have here an idea of the challenge. So the challenge is based on synthetic data, okay? And you have here maybe, I guess it says 10 tomograms composed of 10 macromolecule types with different size. These are macromolecules were randomly distributed in a volume. Here you have the ground truth, and the input is the following image. And the goal is to find all the macromolecules with the appropriate label given from this input image. So the score is to evaluate the performance of each algorithm is based on F1 score, which is a value between 0 and 1. If you have a score, F1 score is 1, is perfect, and that's the idea. So that's why general methods are very close to 0.7, 0.8 as one score. And the same algorithm was ranked very well in different during the 3 challenges without modifying any parameter. It was a good use for us because the backbone finally, this backbone was quite robust for these three challenges, during these three challenges. Okay, what are the results on real experiments? So I said before, the first objective was to perform as well as a human expert to detect ribosome. Actually, this algorithm was able to do the job. We did not outperform the human expert. We just imitate the performance of the expert on ribosome. Okay, but the expert is not able to go further. I mean, for smaller macromolecules, lower than 3,000 kilodaltons, it's impossible for an expert to detect these macromolecules. But in collaboration with another group and at IGBMC in Strasbourg, we focus on nucleosomes. And nucleosome is much more smaller, 200 kilodaltons, right? But to get training data sets, we have to adapt the strategy for training. Actually, an expert, after several rounds of visual inspection, we increase the set of macromolecular for training by inspecting all outputs given by the software called the finder, and the expert select, discard, or keep the outputs to increase the size of the training sets. So by running this iteratively for several rounds, we improve the size of the training sets from 1,000 particles to 10,000 particles. But it took 1 year to get this result. So, but at the end, it was possible to have a better reconstruction of the nucleosome in 3D. And we also analyzed the spatial distribution in nucleosome in nuclei. And we also developed an analytical model able to represent the spatial distribution of nucleosome in the nuclei. Okay, so it could be the end of the story, but recently the Chan Zuckerberg Imaging Institute proposed a new challenge, but on real data set in that time, and the number of participants was much higher than that from challenges, because now we are maybe less than 1,000 people to participate to that challenge. And the annotations are partial in that case. In the previous challenges, the annotations were exhaustive, but in that case, the organizers only provided partial annotation. It means that in some location in the image there is no annotation. Sometimes it's a problem for training. Right. So in the challenge was, is it possible to process each 3D tomogram in less than 2 minutes? Which is not an easy task. Typically, DeepFinder was not able to process such tomograms in 2 minutes, and we have to update this software to compete. So at the end, the best score were obtained by the NVIDIA team. This was not a surprise. They designed a model composed of 70 millions of parameters. Okay, DeepFinder was composed only of 1 million parameters, and they trained several models at the same time. Okay, right. And so the F4 score in that case is 0.8. and DeepFinder was 0.6, right? But at the end of the challenge, we know that the organizer used DeepFinder to annotate the data and to build the training data set, which is a good use for us. And don't remember that Transformer was not able to produce very spectacular results on that challenge. All best competitors used CNN, right? Which was a surprise for us, actually. Okay, so to adapt the definer to that challenge, we modified the architecture. We moved from TensorFlow to PyTorch and so on. We removed the mean shift by a watershed algorithm because it was faster, and we introduced 3D connected component labeling to speed up the algorithm. And now we are able to process a 3D tomogram in less than 2 minutes, which is very nice news for practitioners. Okay, so I will stop down here for the cryotomography, and now I would just want to move how we can adapt this technology to detect very rare events in turf microscopy. The idea is that in that case, maybe you are more familiar with this problem. The idea is to detect very specific events when the vesicle dock at the plasma membrane. And the turf microscopy is typically the right technology to detect these events. So in the past, maybe a lot of methodology have been developed to address this problem in 2D plus time, right? And all of them are more or less based on the following idea. Because we have a pH sensitive probe, I mean that during when the vesicle is very close to the membrane, you observe a peak of intensity, right? And then a decrease because you observe a diffusion in the lateral direction, right? So all the software are based on technology, but some of them are not so easy to use. There is not so the codes are not all codes are not available, and sometimes we have problems with reproducibility of data, and they are all based on very standard approaches. So our collaborators, Christine Schauer and Sophie Massé and Hugo Lachouer, asked us, is it possible to design a CNN method to do this job? And as before, they provided 3D coordinates of these events. So the problem is very similar to the previous one, okay? And here we consider the 3D plus time sequence as a 3D volume. So, okay? And so we apply the same methodology. So, okay, we have 3D coordinates, we can generate mask and so on and so on. We test the method. Of course, we design an appropriate database for different low SNR conditions. Because we would like to be able to detect all these events for many conditions. So we applied this methodology, but it was not successful. If you add if you only consider annotation given by the expert, which is very large, the number of annotations is 12,000 annotation. The definer was not able to detect something. Right. The main reason is that because you have to detect only one pixel in one image, not more. And CNN are not appropriate to do that. Okay, to detect nothing is very hard. So I have to detect very rare events. So but from the experience with the definer, we know that it's easier to detect very rare macromolecules if you consider several classes at the same time. Okay, for instance, to detect proteosomes, if you train a system to detect proteosomes, you never find proteosomes in a cryotomogram. But if you train a system to detect ribosomes and proteasomes, you detect proteasomes. And here we did the same thing. So the second class is spot classes. So we generate, we use a conventional technique to generate spots corresponding, because you have many spots in these images, but only one exocytosis events. So we build a training data set composed of manual annotation and automatic annotation given by a basic spot detector. Right. And the performance will improve. Nevertheless, we have to modify a little bit the strategy. Of course, we generated mask because an exocytosis event can be represented by a small spots with short along the time axis. But we observed that sometimes you never find anything. And we had updated the strategy during the training, because if you consider very large context, we don't find anything. But if you consider very small context, you have many false alarms. You detect many exocytosis events. That's why during the training, we improve, we consider d'abord first small patches to have many false alarms. And after a few epochs, we increase the field of view to guide the model to detect these events. Although we introduce data augmentation, consists just to applying basic operations to increase the data sets and to improve the performance. Right. Okay, so we evaluated this technique on real data sets, and we have the ground truth. So sometimes it's possible to evaluate the performance by computing the F1 score, as here, right? After and before after histaminin. You see in that case, histaminin tends to stimulate exocytosis rate in general. All right, so we observed that before and after there's a conditions, the technique was able to detect satisfying the number of exocytosis rate. And the second, we evaluate the capability of the method to imitate the performance of human, right? And it was quite satisfying here, because you have the two numbers that you have here, right? Tells you that the distance between before and after the number, the number of events is very similar to what DeepFinder was able to produce. So we imitate the performance of this of the expert. And the second step, is the method able to produce satisfying results if you apply the method on another cargo? Because we have many training data set provided for VAM7 fluorine, but we have no annotation for other cargos. So the idea is the method robust to the situation? Is it able to detect event exocytosis in that situation? As you can see, the results decrease, but it's not so dramatic compared to what it was expected. But some work need to do as well. Right. So from these two studies, actually, we draw a few conclusions and perspectives. Yes. First, I told you that it could be a very tedious, long process to acquire annotations. Actually, it's not cheap to get annotations. It took one area to improve of factor 10 the set of training data sets. And that's why we decided to move to simulation. And we benefit from the fact that in the PDB data bank, you have a library of all macromolecules that we can find in the cell. The problem is, okay, we can simulate probably a macromolecule and a subtomogram corresponding to macromolecules. But the most difficult task is maybe to distribute all these macromolecules with the right numbers in the cell, because the abundance of macromolecules is not the same for everyone. Recently in the literature, generative diffusion models have been introduced to simulate images. But if we focus on that case, first it was developed for 2D images. And the problem is that you need a large training data set, which is not in our case. It requires intensive computations, and it may hallucinate. So that's why we resorted to physics and random set simulation to generate synthetic data that can mimic what we can observe in real data set. So we have a library of macromolecules. You can deform these macromolecules by applying molecular dynamics, and then you randomly distribute all these macromolecules, but the right number for macromolecules if possible, in the 3D volume, and then you apply the cryo formation process. That means you have to introduce some correction introduced by missing wedge, plus noise, and so on, to get realistic, if possible, objects, volumes. And then we train Deep Finder on these simulated data. Here you have an illustration of this simulation compared to the real image. And we train this model, our model, to detect in that case. What is the advantage of simulation? Actually, in real data, all macromolecules are not oriented in all orientation. Sometimes you have preferential orientation for macromolecules. So you don't observe anything in some time, in some locations. But with simulation, you can simulate in all orientations. So you can better learn what you want to detect if you apply simulation. So we did that. So the final method is called template learning now. So we compare to template matching, which is the basic approach. And here, just focus on this part of the figure. Actually, template learning provides higher precision and orientation isotropy. I mean that you have particles in all 3D directions. So you can detect more easily some particles that can be maybe found once in the tomogram, because you have a model that can adapt to any configurations, plus deformation and so on. That simulation is very helpful to train a model. The second time, you need just one expert. You can simulate many data if you want. That's a good news. Maybe you need intensive computing, but you save time and probably you save money as well. Okay, second conclusion is that sometimes the model are not able to process data with different brightness conditions. Here, typically, these images are coming from the CZD challenge. So they provided different images. And if you train a model to detect macromolecules here, it probably doesn't work on this image. That's a problem. So the basic approach is either to apply data augmentation, means that you have to generate a large training data set corresponding to all possible conditions. A second step or another approach could be maybe to fine-tuning the model, train on the first condition and to adapt to other conditions. But recently, we demonstrated that you don't need to do that. You need to modify the architecture to improve the generalizability of the model. So in that situation here, we train the model from the worst condition, right? And we evaluate the model with the standard architecture. As you can see, the F1 score decreases very rapidly if the brightness conditions are not appropriate compared to the training data sets. But if you consider a new architecture that is based on equivariance, you preserve the performance of the model. It means that the model, you don't need to retrain the model. The model is robust to different spatial conditions, so you save time and you save money once more. Right, and now to conclude, just a teasing of recent a PhD student will defend his PhD thesis at the end of this year. I presented at the beginning of my talk the result of CellPose. CellPose was developed for 2D images, but CellPose now is available for 3D images as well. Okay, StarDist was also developed for 3D images, but the problem was cell pose and StarDist and so on say the output are binary maps. Okay, so this is a grid of pixels, binary maps, and so you cannot compute curvature from these binary maps. So here we design a new approach that combine CNN with active surfaces. Maybe some of you know the snakes or active contours developed now 40 years ago. Okay, the idea is you can represent a shape by a surface controlled by landmarks or control points, and the shape is approximated by spline, 3D spline. Okay, so here we developed a particular CNN, an hybrid CNN that combine the performance of CNN and active contours, and the outputs of this method is as before binary maps and a set of objects where you have the centroid, and these objects are approximated by surfaces. So it means that now you can compute curvature on these objects, which can be very useful for biophysics. Here you have an illustration of this methodology, which can adapt to different scale and different conditions. Second point, and this method, which is I don't have time to explain details, but this method can be understood as an extension of StarDist 3D and SplaineDist 2D. This is the best of these are two worlds to produce 3D segmentation objects that very helpful for tracking, for instance, in the future. Because today, if you have binary maps, it's not easy to track objects from CNN outputs. Okay. I will conclude here. Just I would like to thank my collaborators from different laboratories in France and Germany, and the members of the teams who participated to this projects. All the three softwares I mentioned here can be found on the different GitHub if you want to play with these tools. Thank you very much for your attention.
Thank you for the talk. I had a question about your the exocytosis data, and I wasn't clear. How do you distinguish from a vesicle that docks and then leaves versus docks and merges with the plasma membrane? Because those should appear identically in TURF or in the fluorescence imaging.
It's not an easy task. Just show you and maybe the video to illustrate this idea. Okay. Actually, we discussed with two experts about the detection of exocytosis. They analyzed the same data set, and the consensus was only 30% of results were okay. It means that it's very hard to identify what is a real exocytosis event. It depends of your probe, of course. Okay, from 7 or another. Of course, it depends on your probe. But from my feeling is that actually we perform this technique. We compare definer to the result of the outputs of the two experts, and the consensus was 30% was okay for each one. It means that maybe in a mind or expert that do not label the events similarly as the others. They are not from the same group. Maybe if you are in the same group, you label the event similarly, but from different groups, probably it could be different. So it's that open question. For sure, that's why I need, as I told several times that you need experts to generate this training data set. Sure.
The training set, yeah. Okay, thank you.
Thank you for the great talk, Char. I was wondering, when you estimate the spline instead of maps for the segmentation,
You mean the last
yes,
slide?
in the gene. So you reduce the parameter space.
Yeah.
So do you get more robustness in detection by doing so?
You mean for the here for this?
Yes.
Okay. I didn't give you details about the methodology here. Actually, the input of the the methods is ground truth, which are binary maps, and your input image. You have to sample the binary maps, and so you have to.
So you still have to go through the higher
Yeah, you have
dimension.
to have a strategy to sample correctly the objects. So you have to distribute the points. You have to limit the number of points as possible. And it was hard to increase the number of points at the beginning of the PhD thesis. But at the end, we had good news that this year, the PhD student found a solution to increase the number of points so you can better delineate the structure you want to detect. I just mentioned here that compared to star disk, you can detect deformable objects with complex geometries. General star disk is limited to star-shaped objects. Okay, so typically an example of complex objects is this one. You have a small curve, a low, high curvature here. Star disk cannot do that. Okay, in general.
Yeah, we can have one last question.
Yes, I have actually two little questions about your simulation of the cryo-ET maps. So the first is, I mean, so the first question is how the learning process is actually influenced by the relative positions of macromolecules. Like in reality, they are not randomly placed. So this could have an influence. And the second question is that the PDB is very biased in the sense that it only represents protein structures that are essentially ordered and that were crystallized. And so how do you take this into account?
For sure, for the PDB, we cannot do anything. So we benefit from this library. We apply here, we deform the macromolecules to improve the variability and to map better to the real data. That's why we apply molecular dynamics to deform the structures. But of course, the starting point is the PDB. It is probably biased, as you said, but it was the starting point. Now, because the objective is just to detect where are the particles and to count the number of particles, macromolecules, maybe this approximation could be okay. The second question that you ask is about how do you distribute these particles. For the time being here, we use a Tetris approach. It means that if you have a macromolecule in some location, you cannot put another particle randomly, it does not overlap and so on. That's just a basic crowded constraint. Okay, but if you know a priori how many macromolecules from one time you have in the tomogram that you can supply this information. If you have 10% of macromolecules are maybe ribosome, if you have only RNA polymerase, only one, maybe you can impose a few proportion. Of course, given this, the outputs of the software on real data can help you maybe to better design how these particles are distributed in the future. So this is an iterative process, I guess. It could be that you can train how they are distributed in this. And for sure, for CNN, it's not appropriate for the time being to do that. So you have to resort to classical approach based on spatial statistics typically.